So let's, I guess, just do a kind of a quick little rundown. So what we're going to do, if you guys have paid attention to your schedule at all, is it says chapters 32, 33, and 34. I'm going to kind of combine 33 into 32. And so anything that you would have gotten in 33, which is a super short chapter, I'll kind of fill you in on with chapter 32. If you don't have your lab manual, again, for whatever reason, that's totally fine. I think I've put everything up on Moodle, including the assessments that you'll need for this part of the lab. So again, if you have any questions, comments, concerns about all that, reach out to me and let me know. But we should be good to go. And please remember, you have an exam next week. And it will be on the nervous system and special senses that we're going over this week. Please do not neglect the couple of histology slides that you have for the last unit. There are no histology slides on this unit. Test format will be relatively the same on Moodle with a bunch of figures that have been uploaded that you will have to label. Okay? Cool. With that being said, let's begin. Again, everything that I show you is going to be up on Moodle for your perusal, but just so we're all on the same page, right? This document is up on Moodle. It just says, hey, chapters 32, 34. These are what you have to label. On the one that I loaded on Moodle, I just caught this mistake. It said 32.2, 32.2, so it's 32.2, 32.3, right? I've fixed this one, so just maybe make a note of that. Assessments A and B, which are on Moodle. Then chapter 34, label 34.1 to 34.3. And then also towards the end of the chapter, and again, we'll go over this one together, but I don't want you to forget about it. This is figure 34.10, and this is the figure that you would have used to identify the different structures if you would have been dissecting a eyeball in person. All right. So please just don't forget to look over that. And assessment A for that chapter is also on Moodle, right? So nothing too crazy there, I think, for you guys. Okay. So with that being said, if you have your lab manual and you want to, you can go ahead and turn to 32.1, which should be a picture of the ear. I'm going to pull that up so we can talk about some of it. I'm not going to give you every thing that you need to label. Some of you have probably already started, which is awesome. Maybe some of you are finished, which is even better. But those of you that haven't started or aren't finished, right, you'll have to go back and do some of this on your own. All right, so let's pull up that first image that I want you guys to look at. And of course, I did not have it pulled up front. Okay. And shoot. All right, stop the share. Sorry, I couldn't see it. All right, now I'm on the same page. All right, so this is the ear, and you'll find out, if you haven't already, that the ear is divided into kind of three basic regions, right? And you just kind of start from the outside and work your way in. So you have the outer ear here. So if you think about pretty much anything you can touch without causing pain, right, that's going to be your outer ear. You then have this little pocket within the temporal bone right here. This is the middle ear, and it houses a couple small bones that are important for your ability to hear. And then you go to the inner ear, right, that contains something called the cochlea, which is this kind of snail-like structure right here. If you haven't labeled it, that would actually be number 6. That's the cochlea. All right, this is going to take neuronal impulses that are generated by sound waves and process them in your brain to turn it into sounds and things that you understand and recognize. And then you have this kind of structure right here that looks like it has these couple loops. That would be number 4. These are the semicircular canals. Does anyone know, or can you tell me what else the ear is for besides hearing? What else is important for?
Balance.
What do you say, Bruno?
I said balance.
Balance. Perfect, right? Bruno is right. So if you've heard someone talk about the ear, when they talk about equilibrium in the ear, right, they're referring to its ability to help you maintain balance, all right? And we'll talk about how that works here in just a little bit, okay? Let me check on my sheet here. It is here. Okay, good. And Avery's here. Perfect. All right. So let's kind of start with the outer ear. We'll talk about the middle ear and the inner ear, kind of in that order. So I'll give you a couple of these, right? Some of them you have to look up on your own. Number 1, has anyone already done the labeling? Can you do the thumbs up thing on your thing if you have? Avery, Abby, Lauren, Hannah, Peyton. Bruno. Okay, good. So you guys know a lot of these. That's awesome. So I'll kind of lean on you guys a little bit for some of them, but then we'll still kind of skip over a few of them. So Hannah, what's number 1? You had a thumbs up, right? I hope. Hey, babe, can you pull that door shut? Because someone came.
Yeah, it's the oracle, right?
Yeah, the oracle, also called pinna. All right, cool. So if you look at that, and then it's connected to number 11 here, which is the external acoustic meatus, right? So some of you probably already have that labeled. If you don't, write it down quick. If you can't get it down, then go ahead and look it up later. And I believe this is chapter 12 in your textbook that will have all this information and all these answers for you, okay? So the oracle, along with the external acoustic meatus, what are they for, right? And I understand maybe you didn't look up the function, but maybe you did when you came over it, but just kind of look at the shape. And what do you think that part of the ear is for? Anna?
Repeat the question again, I'm sorry.
That's okay. So if you look at the oracle, which is number 1 here, along with the external acoustic meatus, which is number 11, what do you think, based on the structure of this, right? What do you think the function is?
Directing the sound. And if not that, I know that the number 11, they use that for kind of protecting the eardrum as well.
Okay, perfect. So, yes, so primarily, right, I'm going to go with, I like your first one. Your other one's not wrong. Right, when we're talking about the physiologic function, Hannah is right. Wait, was that Hannah or Anna? Who did I just ask? I asked Anna. Anna was right. I got you, Anna. I won't leave you behind, right? So Anna's right? It's going to help collect and funnel sound. So if you actually look at its shape, it does kind of look like a funnel, right? And then it leads to number 9. Lauren, what's number 9 here? Lauren? Bruno, help me out. What's number 9?
Tympanic membrane.
Perfect. The tympanic membrane. For those of you that are band oriented, maybe you played band in high school or middle school, right? A timpani, right, is a really big drum. I mean, they have like huge drum heads and they kind of have those cool like copper bases, right? And so basically the way that drums work to create sound is their heads, the drum heads are really hard and tight, and you hit on them and it causes a vibration, and that vibration is amplified by the body of the drum. And that's essentially what you see going on here in the ear. So sound waves, right, hit the tympanic membrane and they cause it to vibrate back and forth, all right? They then pass that vibration onto 3 small bones, which are represented by 2, 3, and 5, okay? The collective name of these bones is the auditory ossicles, all right? The auditory ossicles. And you, right, will have to label for me, even though I'll actually go over some of them and just a little bit, right, what those auditory ossicles are for me. Please remember, if you have me for lecture, one of the things that we talked about was osteogenesis imperfecta, where those patients have brittle bones because they have a mutation in the collagen gene. And so their bones are hard, but they're not strong. And some of those patients actually have trouble with hearing. And the reason they have trouble with hearing is because these bones don't function properly, okay? And so these bones in the middle ear pass the vibrations from the tympanic membrane to the cochlea, right? Where they can then be turned into neuronal impulses, the sound waves that are processed by the brain and become the sounds that we know and hear, all right? So we didn't label all of those. We labeled a lot of them. You'll be responsible for the ones we didn't. And actually, one last one before we go. Avery, what is number 10?
The auditory tube.
The auditory tube, right? Also called the eustachian tube, all right? And so who can tell me what the auditory or eustachian tube that Avery just pointed out is for? Grace, are you there?
I'm here.
So what's the auditory tube for?
Oh.
Grace, have you ever flown in an airplane?
Yes, I have.
What do your ears do?
They pop.
They pop, all right? And the reason they pop is when they pop, that is actually you equalizing pressure, right? That's in the cabin of the plane with the internal pressure, right? And the reason that you want these pressures to be equal is it helps the tympanic membrane remain tight, right? And so it can receive the vibrations properly. Those of you that have flown, right? Before your ears pop, you may have noticed that sound is kind of muffled. You don't hear quite as well. And that's because you don't have the proper tension in your tympanic membrane. And so when the sound waves hit it, the vibrations, right, are not as strong. And so you can't hear as well. So when you pop your ears, right, it equalizes pressure, right, and you get the proper vibration. Now, what it doesn't show down here, right, is the pharynx. Oh, the pharynx is a fancy word for what, Beck. Don't overthink it. Your throat. Good job, Beck, right? Your throat, right? So these tubes are connected to your throat, all right? And what it doesn't show right here are 2 kind of little flaps of skin that cover them. So when you're flying, or maybe you've, like some of you have hiked and there's a big elevation change, and maybe you chew gum, maybe you yawn, all right? That will help move that will help move those flaps and allow air to go in there to equalize the pressure between the 2 sides of the tympanic membrane. All right. So that is the ear, right? You need to kind of go back and finish whatever you don't have filled in. Okay. All right. So let's go to your next image. Perfect. Here it is. So now we've kind of made our way into the inner ear. And here's the cochlea that we already labeled, right? Which was actually, I think, number 6 on the last figure. And it's also number 6 here, so you can put number 6 here. Number 1 here, you can see there's 3 of them. Here's one, and here's one. These are the semicircular canals. All right. And then 2 and 3 here, right? 2 is going to be the utricle, and 3 is going to be the saccule. All right. And it's the utricle, the saccule, and the semicircular canals that are important for equilibrium and balance. All right.
Hey, Dr. Deft.
Cole, you made it.
Hey, yeah, I'm on a short lunch break, but
Okay.
I managed to be able to get on, so I'll probably only be on here for maybe 20, 30 minutes.
That's fine, because I know you couldn't make it, and we have a couple other people that can't, so I'm actually recording this one. And what I'll do is I'll put it on YouTube and then send you guys the link. So I'll send you the link, Cole, and then the couple other folks that I know had to miss because of tornado-related reasons. All right, so you'll be able to see everything we did, okay?
Okay.
All right.
Awesome. Thank you.
Enjoy that lunch. So again, equilibrium and balance in this part of the ear is actually going to be for hearing. Now, one of the things that I want you to know a little bit about is maybe a little more detail on the function of these different components. And what I want you to realize is that you may need to go into your textbook to read up on these very briefly, right? I'm going to give you the details that you need, but you may not. Okay, so some of you are having some issues. Okay, well, that's fine. Like I just told Cole, I hope Hannah and Peyton, I just saw you're having some issues. If you can hear me, that's cool. I'm recording this one, and so what I can do is I can make it available to you guys if you're having issues. So try to hang with me. And Peyton, every time I call your name and you don't answer, that's only 5 points off as opposed to the 10 that it was before, right? So that's me being reasonable. All right, cool. So let's talk about equilibrium first. Okay. You're welcome. You're welcome. You're welcome. All right. So for equilibrium, there's 2 types of equilibrium. There's static equilibrium, and there's something called dynamic equilibrium. I realize that may be more than you guys want to know. This may be more than you want to know, but I think they're important terms to kind of throw out there and realize that although the utricle, the saccule, and the semicircular canals are all contributing to equilibrium, they all kind of do it in different ways, right? So let's talk about the utricle and the saccule first, all right? So number 2 and number 3, all right? So these are going to be important for static equilibrium. What that means is they're going to help you maintain proper position of your head and maintain proper posture while you're sitting or standing still, all right? And they do this with the aid of these little things here called maculae, right? So it shows a couple of them. So maculae is plural, macula is singular. So what happens, right, in the utricle and the saccule is that they have these little things called hair cells. And hair cells are little cells that look like they have little projections of hair coming off of them, all right? And the hair from these cells is embedded within these maculae, all right? And the maculae are kind of like a gelatinous jelly-like substance that has something in it called calcium carbonate. And calcium carbonate form little crystals. And those little crystals are important because they make the maculae kind of heavy. They give it weight. And so when you move your head, when you're sitting or standing, it actually causes the maculae to move slightly, to kind of slide to the side a little bit because of the calcium carbonate crystals that are in them. And so when that happens, when the maculae move, right, they rub on the hair cells, and those hair cells are connected to the dendrites of neurons that then feed back into the brain and allow you to process that motion that you're experiencing. Okay? So that would be those two sections right there. Let's talk about the semicircular canals next, and then we'll move into the cochlea. So you can see you have 3 semicircular canals. The relationship between all of them is at 90-degree angles. Through the semicircular canals, you have a fluid running that's called perilymph. All right? And this perilymph is going to rush over cells that you would find in section number 4. So at the end of each semicircular canal, you can only really, well, you can see it actually right here. See it kind of bulges, it looks like it's swollen a little bit. And that's at each semicircular canal, and we call that an ampulla, all right? Within the ampulla, right, you have some specialized hair cells, again, that are embedded, all right, within a jelly-like substance, right, that we call a cupula, C-U-P-U-L-A, all right? And so this cupula forms this triangle of gel, and embedded within that triangle, you have these hair cells. And so when you move, fluid, in this case perilymph, rushes through those semicircular canals. It hits against that cone of jelly, and it causes it to move, and that stimulates the hair cells. And then that sends a signal to neurons that are connected to your brain. Now, when you move in all sorts of different directions, right, different amounts of fluid are moving through these canals. And so your brain is able to process how much fluid is moving through which canal, or how much each ampulla is being stimulated, in order to determine which direction you're moving. And to kind of put this in perspective, right, if any of you have done the, I don't know, like in a relay race when you were a kid, or maybe somewhat more recently for whatever reason, you do the thing where you put your head on the bat and you spin around 10 times, and then you try to run, right? And even if you stand still, right, things seem like they're spinning. And the reason that happens is because when you spin that quickly, you're moving faster than the fluid can keep up with. And so once you stop moving, although you've stopped, the fluid in those semicircular canals is still moving and stimulating those ampullas. So even though you're standing still, it feels like you're in motion, right? So that's what's going on there. Cool? Any questions, comments, or concerns up to this point? Abby, you good?
Great.
That is what I like to hear. I'm glad for that. All right. So let's talk about the cochlea. Now, this image of the cochlea, I think, is in, I can't remember if I put this one in the PowerPoint that I put up for you guys, but there is a PowerPoint on Moodle that has some of these other images. So please be sure to check out that PowerPoint while you're looking at the other images. So what I want to show you here, right, is what the cochlea looks like when it's unwound, right? So I'm sorry if this image is a little confusing. This is actually the same structure with 2 different views. So here is the wrapped up cochlea that looks like a snail shell that you saw in that first image that we labeled and in the last image that we were going over. In this case, what it's showing you right here, though, is what this looks like when it's unwound, all right? So we're kind of imagining if we unwound the cochlea. And there's a couple of things that I want to point out, right? You essentially have 2 chambers in the cochlea. And in this case, right, this top chamber, the scala vestibuli, right, which again is filled with perilymph, much like the semicircular canals. This is where sound waves enter via vibrations. And those vibrations are coming from that 3rd auditory ossicle called the stapes. So this is one of those small bones in the ear. So connected over here, you'd have another small bone, and connected over here, you'd have another one, because remember, there are 3 auditory ossicles, all right? And so the stapes, which vibrates because of sound waves that originally hit the tympanic membrane, all right? It vibrates, and it's connected to something called the oval window right here, which is the opening that leads into the scala vestibuli. And those vibrations are now moving through this chamber, right? So the vibrations move in the directions that you see these arrows right here, these kind of dark blue arrows. Now, one thing that I want you to appreciate is that running through the middle of these 2 chambers, I'm sorry, the bottom chamber, right, is the scala tympani, right? So you have the scala vestibuli, the scala tympani. So vibrations enter the scala vestibuli, they move through, and then they enter the scala tympani, boom, 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 before they exit. And we'll talk about their exit here in just a second. But separating this chamber is something called the cochlear duct, all right? And the cochlear duct, right, the outside of it is represented here in purple. So you can see it's hollow on the inside, and we'll talk about some of the structures that are inside it in just a minute. But what I want you to appreciate is that as sound waves are moving through, they are bouncing, right, inside of this chamber and rubbing and bouncing up against this cochlear duct, all right? And then when sound waves are done, right, when they reach this side, they'll actually hit this area called the round window, and the round window stops the sound waves. All right? Why do you want to stop sound waves? Does anyone know? Have any of you ever done like worship team at your church or band or something, and you have feedback that goes through a mic? Right? What does that feedback sound like? Has anyone heard that before? No? No feedback? You guys haven't experienced feedback? You haven't lived yet, right? Feedback basically is when sound comes out of a mic and it's really loud and screeching and unpleasant. And that kind of makes sense, right? Because a mic is where sound is supposed to go in, not come out. So this round window absorbs sound waves so that you don't have those sound waves going back and coming out the ear. That would really confuse the nervous system, and you constantly have this screeching and not be able to process the sounds that you hear. Okay, so let me erase that. Perfect. So what does this look like inside this cochlear duct, right? So these sound waves are hitting the outside of the cochlear duct. How are they leading to generating a neuronal signal that results in the sounds that we all know? So if you have your lab manual, you can turn to 32.3, and I actually forgot to load this image separately, but this is actually 32.3 right here that I put in here for you guys to practice labeling. All right. So just a little quick orientation, right? We're looking at the cochlea. Again, this is the scala vestibuli. This is the scala tympani. All right. And remember, sound waves are running through there. And when those sound waves run through there, right, they're hitting up against the cochlear duct. All right. And inside the cochlear duct, you have this structure, which you see in 32.3 if you have your lab manual. And so there are a couple of different features of it that I want to point out. Now I want to make some thicker lines. So here, right, you have this structure, right? This is the tectoral membrane. I'll talk about what it does in just a second. You also see down here a group of hair cells. And if you look closely, you can see the little hairs that are basically almost touching or are brushing up against the tectoral membrane. You also have a cochlear nerve right here. And please note that that cochlear nerve is attached to other neurons that are running from the hair cells, right? So they're all kind of gathering there to make one big nerve or cable. Okay. And you can actually see that here, all these little kind of spaces that it collect and converge there. And then let's see, what else do you guys have to label? Hair cells, okay, yeah. And then just down here, the bottom of it, this is just called the basilar membrane. All right, so again, you guys have heard of the basal side of epithelial cells. That's all that's going on right there. So how does this work? All right, so the sound waves are moving through these two chambers, all right, the vestibuli and the tympani, all right, and they're kind of bouncing off the inside of those chambers. When they bounce off the inside of those chambers, it actually causes the tectoral membrane to shift back and forth, all right? So it starts moving. When it starts moving, it rubs the hair on the hair cells. When the hair on the hair cells is rubbed, that sends a signal to the neurons that are connected to them. Those signals are collected and then sent to, right, somewhere within the temporal lobe, right? We mentioned that, I think, in last lab, right? Now, who's heard of a word that reminds them of tectoral, right? So this is the tectoral membrane. Oops. Bring that back. Bring that back. Come here. My bad, guys. What's another word that reminds you of that, where things move back and forth?
Tectonic plates.
Perfect, tectonic plates, right? So those of you, right, and occasionally does happen here a little bit, rarely, but if anyone's kind of from the West Coast, right, maybe more of an everyday part of life. Tectonic plates, right, exist within the earth, and when those move back and forth, that's what causes an earthquake, right? So that might help you remember this tectoral membrane. It moves back and forth, it stimulates the hair cells, which are connected to neurons, which send information to the brain, which allows you to essentially turn those vibrations into the sounds that you guys know. Okay, we're good there. Let me erase this, and then we'll keep on chugging along. Oh, and also just kind of like a little interesting side note, right? Over time, all right, that tectoral membrane, right, as you just get old, right, imagine if you have a grandparent or a great-grandparent that's in their 70s, 80s, 90s, those hair cells eventually die, right, from just being stimulated and overworked. And this is one of the reasons that older folks have trouble hearing. And unfortunately, right, these hair cells don't grow back. So once you lose them, that's it. This is also a reason for those of you that maybe love to go to concerts, right? You'll notice some people that are closer to the front, right, they'll have their earplugs in. Intense stimulation, right, can shorten the life of these hair cells. So if you're someone right now that's going to a rock concert every week, or your favorite concert, whatever type of music you like, and it's really loud, and you do that over and over and over again, well, yeah, then your hearing might go a little bit earlier than some of your peers because you've stimulated your hair cells more than that. All right, so that's kind of what's going on there. So for the rest of it, you have to do assessments A and B, which I do have up on Moodle. If you have your lab manual, or you can look at this in your textbook, but in your lab manual, it's image 32.4. You can see all three auditory ossicles are laid out on a penny, and they only take up about 25% of that penny, right? So these bones are very, very small. So go ahead and finish labeling anything from that unit that you haven't done yet after we get off of this Zoom session, okay? So assessments A and B here. Chapter 33, again, I said we're kind of skipping over it because it talks about the ampullae that you find at the end of the semicircular canals, which I kind of described to you guys. If you want to see a little bit more as to what that looks like, you can look at 33.1 in your lab manual, or if you don't have your lab manual, you can go to your textbook and look up an appule at the end of the semicircular canal, right? So let's then go to chapter 34, okay? And this is the eye. What we're going to talk about are some of the we'll say structures external to the eye, but are still important in eye function. So I'm going to go ahead and let's see, arrange those in the way that I want. Here we go. So the first one I want to show you guys. So again, this image is up on, how did I lose it? Oh, there it is. All right. So this is showing you a couple different things here, right? And maybe you're like, why are we looking at this? What's the eye? But I promise this all kind of makes sense. So if you're looking at this image, we're going to start up here and then we'll work our way down. Let's see, do I want this? I'm trying to look at one way. There we go, perfect. So what you see here, all right, is a gland and a series of canals that are connected to the eye. Does anyone know that's already labeled, or can they tell me what that gland is?
The lacrimal gland.
The lacrimal gland. Good, awesome, thank you, Peyton. So in this case, the lacrimal gland, right, is kind of situated kind of right here. You can actually, when you rub your eye right or above your eye right here, you're actually touching your lacrimal gland, all right? Your lacrimal gland produces what, Seth? Seth, you there? All right, we'll come back to my boy Seth. Someone help me out. Hannah, what does the lacrimal gland produce?
Tears.
Tears. Perfect, right? And so what we don't really appreciate is that every time you blink, you're helping, right, spread the tears that are produced by the lacrimal gland over your eye. And so it's actually meant to keep your eye moist. So you're generating tears all the time. If you go to number 2 here, this is the superior caniculi. This is the inferior caniculi. I don't know if this helps or not, but if you were to press through right here on your eye, right, you would hit the lacrimal bone, right? That's one of the bones that you guys have to know in the skull. So that bone is situated right beneath this tissue. Lauren, can you remind me where we've heard the term caniculi before? Lauren? Avery?
I don't remember.
You don't remember? Okay, that's okay. Cool. Grace?
I don't remember.
Bruno?
You said caniculi,
Yeah. Do you remember what unit we talked about that in before?
the bones.
The bones. Perfect. Good. Thank you, Bruno. If you remember, those osteocytes that exist within the bone, right, are connected by these little small canals that kind of look like little cracks in the bone. Those caniculi allow the sharing of nutrients and the disposal of waste that are generated by osteocytes. All right, so here again, these are small canals. And so, right, in a normal situation, if you have any excess fluid, and there might be a little, it's drained through these caniculi and then goes through these series of chambers, right? I will let you look up what these chambers are called on your own. So let's talk about tears. When you cry, all right, let's see, who probably cries the most here? Well, I have some guesses, but I won't go with that necessarily. But Anna, when you cry, what are some of the things that happen with those tears, or kind of things that you experience physically?
I mean, they well up on the bottom lid of your eyes, and then it also like causes sinus drainage.
Perfect, right? That's right on the money, right? And so when you start to cry, like cry, and again, it can be multiple reasons, you start to produce more tears than the caniculi can keep up with. So they start to well up here and then start to run down your face. Meanwhile, right, you're still trying to drain tears, but as you keep on draining, right, you eventually fill up, right, this nasal cavity, and so you might start to get a runny nose, all right? And so that's why, right, when you cry, it runs down your cheek and gives you kind of a runny, snotty nose, right? Because your body's trying to drain and keep up with those tears as best you can. Now, one other thing I want you to make note of, within your tears, you have something called lysozyme. Lysozyme is an antibacterial enzyme that helps keep infections out of the eye, right? The eye is very important and sensitive. You don't want inflammation in it. Inflammation in the eye can cause a lot of serious damage because, again, it's enclosed by bone on 3 different sides. And so by killing any bacteria that might be in there via the tears, it really helps reduce the risk of inflammation in the eye. Special side note too, research has shown, which is kind of interesting, and I need to find the paper on this to look up the exact composition, but the chemical composition of tears varies depending on why you're crying, right? So a happy cry versus a scared cry versus a sorrowful cry, right? The chemical makeup of those tears is actually slightly different, right? Cool. Okay, so let's move on then to the muscles of the eye. Now, I'm not going to tell you what these muscles are named, right? I'm going to let you look that up. But again, if you go to chapter 12 of your textbook, there's a nice diagram with this labeled and then a nice little box that tells you what these muscles do. Suffice to say, these muscles are allowing you to move your eye from side to side, right? Medial, lateral, and then up, down, right? Inferior, superior. And you guys have all experienced this because if you've been to the eye doctor or your regular doctor, right? And they hold their finger in front of you and they move it and they ask you to follow the movement without moving your head, all right? You are using those muscles to position the eye to follow that object, right? So again, up, down, right? Superior, inferior, side to side, lateral to medial. That's what those muscles are doing. Now, so I want to throw out a small extra credit opportunity that will appear on your lab exam. So if, right, you have some time and are interested, go to your textbook, right? And when you look it up to help you label these muscles, you will also see that it talks about the specific direction that they move the eye, right? So one muscle may move your eye up and towards the midline, right, or up or medial. Another muscle may just move your eye down into the midline. Another muscle may move your eye away from the midline. Another one may move it just towards, right? All that information is in your textbook. And if you learn that information, right, I'll tell you right now exactly what I'm going to do on the exam. I will say, okay, label 1 through 6. Not letting me move that. There we go. Yeah, 1 through 6. And then I might say, okay, maybe I'll choose 3 of those muscles. Maybe I'll say 1, 3, and 5. All right, so label 1 through 6, and then for muscles 1, 3, and 5, tell me how it's moving the eye. And if you do that correctly for each of the 3 that I asked for, I'll give you 3 extra credit points towards your exam grade, right? You don't have to. Understand if you're overwhelmed, don't worry about it, but at least learn the muscles. And then if you want a little bit of bonus, go ahead and tell me which way they help the eye move. Any questions, comments, concerns there? Okay, cool. Go to our next image. So if you have your lab meeting, what we're going to do now is go to 34.3. And again, I'm not going to go over all of these. I will point out a couple of these and point out some of their important functions. I do want you to know the functions because they will be important for the assessment that you have to do at the end of this chapter. So this is 34.3. Again, if you don't have your lab manual, that's fine. This is on Moodle for you guys to look at. The eye, like the ear, is divided up into three kind of areas. And so if you start on the outside of the eye, we call that the outer or the fibrous tunic. Once you move in, right, we call that the middle or the vascular tunic. And then even farther in from that, you have the inner nervous tunic. All right. And if you're wondering, yes, the names kind of indicate some function. So the middle or vascular tunic, right, there's a lot of contact with blood vessels that are supplying nutrients for the whole eye that are in that layer. The inner tunic or the nervous tunic is composed of special cells called photoreceptor cells that interact with neurons that generate the signal that allow your brain to process what you're seeing as an image. All right. So let's start with a couple components of that outer tunic. And again, we're not going to go over everything, but I will point out some important ones for you guys. All right. So let's start with number 6, right? Number 6 is the cornea, right? And the cornea allows you to take light that's all around you and focus it, all right? What part of the microscope took light that you were shining from the bottom of the microscope and helped you focus that light onto your specimen? Does anyone remember? Come on, someone help me out. What part of the microscope helped you focus light onto your specimen? I got nothing but time. Ooh, maybe someone typed one. Okay, no, not the lens. It is a type of lens, right? Specifically, it was called the condenser, right? The condenser is what helped you focus light on your specimen. And that's what the cornea is doing. Now, the cornea, interestingly, is made out of collagen, right? And typically, when we think of fibers, we do not think of things that are clear. But in this case, the collagen fibers have a unique arrangement that allows them to appear clear, and the cornea also lacks vasculature, so there's no blood vessels that run through it. All right, so this is both of those things are important in allowing light to pass through it and be condensed. The last part that I'm going to talk about in the outer tunic is, I believe, number 11 here. You can see it's a little farther down from my arrow. And number 11, all right, is the sclera of the eye. All right, so this is the white part of your eye. So if I were to say touch your eye, this is probably one of the first parts that you would actually touch. All right? And the sclera is important for a couple reasons. It protects the structures of the eye. Okay. And it also serves as an attachment point for those muscles that I told you about on the last figure that you guys are responsible for going to look at and label on your own. All right. So the cornea and the sclera, part of the fibrous outer tunic. All right. The middle tunic has multiple components that are important. Again, we're just going to go over a couple of them. Excuse me. The first is going to be number 10. Number 10 is called the choroid coat. And the choroid coat is composed primarily of melanocytes. Avery, what do melanocytes do? Or what unit did we talk about melanocytes in? Abby?
Is that what helps give your skin color?
Okay, it does help with skin color, so it provides pigment. All right. How do those pigments provide protection? Avery, I did say your name. How do those pigments provide protection? Those melanin pigments, what are they doing?
Could it protect your eye from like UV rays in the sun?
Perfect, right? So melanin, right, absorbs UV light. So it provides protection when we talk about your skin. Now, in your eye, what's happening is it's absorbing any excess light and it's keeping the inside of your eye nice and dark. Okay. The way I kind of look at like to think about the inside of the eye is if any of you have ever developed photographs, if you ever took like a photography class, maybe in high school, I think they probably even have one here at Lee, maybe, I don't know. But you've heard of the dark room, and dark rooms are important for images developing. And the inside of the eye is kind of a similar idea. You don't want excess light, and so you absorb it, and it helps you develop and form the images that you guys see. All right, so that is number 10. Let's see, some other ones that are, I think, kind of cool. If you look at 3 and 4, 3 is the ciliary body, 4 is the suspensory ligaments, and they are going to apply pressure and tension to the lens of the eye, which is going to allow you to focus on images. All right, and so that's right here. There's some other stuff to label, but I'll let you guys look over that yourselves. So what I want to talk about next are a couple structures within that inner tunic. All right, and I forgot to mention too, on the choroid coat, and I saw this here in my notes, I wanted to mention to you guys, this is where blood vessels are penetrating the eye to deliver nutrients and to pick up waste, right? So the choroid coat is important because it, right, darkens the inside of the eye, but it also is the point of blood vessel penetration for the rest of the eye tissue. All right. So let's talk about that inner tunic. All right. So the inner tunic, again, has multiple components. The biggest one that I want to talk about, though, is going to be number 9 right here. And you can see it's represented by that yellow here. And that is your retina. And your retina, right, is composed of a thin layer of photoreceptive cells. So those of you that have had maybe biology before or anatomy physiology before in high school, maybe even in middle school, I don't know, you may have heard these photoreceptor cells. There are 2 major types. There are rods and cones, right? We'll talk about those in just a little bit, all right? But you can see that that's this inner layer right here. And these cells, right, process, right, what you see and send that as a neuronal signal to the brain, right, which allows you to kind of form and comprehend the images, right, the things that you're seeing with your normal vision. If you've heard of someone that's been in a severe accident and they have a detached retina, that requires immediate surgery because this is a very thin cell line. And if it's not fixed quickly, that patient will actually end up going blind, right? And you'll see what that looks like here in just a second on the dissection, all right? What you see here, I'm just going to point out 2 more things. 13 kind of looks like this little indentation. That's called the fovea centralis, right? And the fovea centralis, right, is the point of the retina where the sharpest image can be formed. It also belongs to a bigger area, this whole area around it. It's called the macula lutea. So if you've heard of someone with macular degeneration, right, this is an area that's affected severely, and it basically takes away their ability to see things clearly. And most of them are usually declared legally blind because they can only make out like small shapes. There's no crispness to their vision, right? The last thing that I'm going to tell you guys about is the optic disc. And you can see there are 2 major things that happen here. This is how blood vessels first enter the eye through the optic disc. And you can see leaving the optic disc connected to the retina is you actually have a series of nerves, right? And these nerves, again, are going to connect back to the occipital lobe, where they will process those signals and turn them into the things that you see. Ooh, the last thing that I wanted to point out before we move on from this is number 12. Number 12 is actually trying to get you to mention the vitreous humor. So those of you that have dissected an eye before, right, this is kind of like a jello-like substance, and it's actually what gives the eye its structure. If you were to essentially poke a hole through the sclera, so go from that outer tunic to that inner tunic, right, you could actually deflate the eye, and so they just have to close that wound to allow new vitreous humor that's being formed to help fill the eye back out, right? Or back up, sorry. All right. Any questions on that? So there's a couple things that you still need to go and label and look at the functions, and that will also come through in the assessment for this chapter. Okay? Cool. So, I'm sorry, I forgot to mention, if you have your lab manual and you go to 34.4, it actually shows you a microscopic image of rods and cones, right? And for rods and cones, right, they're both photoreceptor cells. So they're both processing information and turning it into vision, which is a couple important distinctions. So rods are really, really sensitive cells. And what that means is that you don't need a lot of light to stimulate them, all right? But the thing with rods is they don't give you like a sharp image. They kind of allow you to see the general outlines of things, right? So they allow you to see in little light, but they don't allow you to really see that crisp of an image. That's where cones come in. They're not nearly as sensitive as rods, but they do allow you to see in color, and they allow your images to be sharp. So you guys are probably not surprised if any of you have been like, I don't know, camping, or you've been up really late, and maybe you have to get up late to go to the bathroom, right? You notice that you still can see in the dark a little bit, but that what you see is not very clear. You're seeing more of just the outlines of things, right? You can kind of tell something's there, but you can't see it great, right? That's because of your rods. When light gets turned on, right, then, right, it's able to stimulate your cones more efficiently, or not more efficiently, more effectively. When those cones are stimulated, right, with that light on, then you can see colors and you can see a crispness or a sharpness to that image. All right, so again, that's all in the retina. So, last image, and then I will let you guys get out of here. I went kind of quick, but that's okay. All right. So this image right here, actually, it's better when it's smaller with you guys. Yeah, this image right here is what you would have seen if we would have been able to dissect an eye in person, right? So this is a cow eye. We might have done that. We might have done sheep eyes, which are almost exactly identical, just a lot smaller. So I just want to point out a couple of those important structures that we've already talked about. Here, right, you can actually see a little bit of the outside of the eye. That would be the white part, right? That's the sclera. On the inside, you see the choroid coat. Now, the choroid coat, if you look at it right here, it kind of looks black. You can see some more of it right here. It's black. That's from the melanin produced by the melanocytes, right? So that's going to absorb excess light and allow better image development. What's really cool in the cow eye, right, and you also see this in the eyes of other animals that are maybe active at night, is something called the tapetum fibrosum, right? It says iridescent. And if we actually had a real eye and do this, you'd see it's this really cool kind of shiny teal, aqua green color. And what it does is it allows light to actually reflect and bounce back and forth in the eye. And this is important because at night, there's not a lot of light available. And so what this does is it basically amplifies the light that's inside the eye. And that's why these animals can see better at night. And you've probably seen this if you've been driving late at night and you see cows in the field on the side of the road, or if you see deer on the side of the road, their eyes will glow when cars come by, and that's because light is bouncing off of this tapetum fibrosum, this iridescent layer, right? Same idea with your dogs or your cats. When you come home and it's dark and you open the door and you don't think there's much light, but their eyes still look like they're glowing, all right? That's from light being bounced inside of this layer to help those animals see more clearly at night. What you see here, and it's detached, it's kind of peeled away, but this is actually what the retina looks like. You can see, or I think you can maybe see, or I hope you appreciate how thin of a layer it is. So that contains those photoreceptor cells, those rods and those cones that allow you, or that interact with neurons, and then eventually those signals result in the sights that you see. Down here, you can see a little bit of it, doesn't maybe quite do it that much justice, but this is vitreous humor, that gel-like layer. All right, and this gel-like layer, again, fills up almost the entire eye, giving it shape and structure. Here you have the lens. If any of you have dissected an eye before, you'll note that the lens is actually pretty hard, all right? It's a pretty hard structure. Now, when it gets treated, right, it kind of changes the color, and so a lens, right, from a recently living organism or a living organism is going to be much more clear than that. Again, it's the protein arrangement and the special proteins within the lens that allow it to be clear, even though it's quite fibrous. What you're looking at here is the inside of the front part of the eye, or the anterior portion of the eye. And so here you can see it kind of looks a little fibrotic, this fibrous tissue. This is the cornea. Right, what you're looking at here is the back of the cornea, right, because you're looking inside the eye. Here you have the iris, right? This is your part of the eye that is colored. And remember the iris diaphragm on a microscope. You can open or change, in this case, the size of the pupil opening to allow more or less light in. Kind of around it in this ring-like pattern, right? You have the ciliary body, and remember the ciliary body via the suspensory ligaments are attached to the lens and can kind of pull that lens in different directions to help you focus. All right, I think that's it there. So just a couple of quick reminders before I let you go, right? All this stuff is up on Moodle for you to practice, right? So go ahead and start looking at it. Your lab exam is going to be next week, all right? On Moodle, same like we did before. Please remember to zoom in. For that, I expect, right, to be able to communicate with you immediately if I need to. It also allows you to ask questions of me if you need them. So please be on top of that, and let me know if there are any issues, or if you foresee an issue coming up. Also, please remember, right, it's this special senses stuff, ears and eye, but also it's going to be the nervous system that we did last week. And one of the big things there is there's a lot of things to label, and don't forget you had 2 slides. I understand you're like, oh, that's only 2 slides, but there's a good amount of stuff on each of those slides. So please remember to look over those and ask me questions. If you have them, I will be more than happy to help you out and answer them. All right. Any questions before I cut you off and set you free?
Well, we have our lecture exam on Friday.
Yes.
Okay.
That's my plan. And I need, if you're a Hobbit, go ahead and check in with her. But yes, the plan is for you guys, you will take your exam, Tuesday, 11:45. And then for my students, and I'm assuming Professor Hobbs' students also, they'll then take your lecture exam that Friday.
Okay.
All right. Because next week is the last full week of classes. And then we go into Monday and Tuesday class with reading day on Wednesday, right? So reading day is 2 weeks from tomorrow. All right. Any other questions, comments, or concerns? All right. If you have something, go ahead and reach out to me via email. Again, remember, we can meet via Zoom to go over this. You just got to let me know what I can do to help you. All right. It was awesome to see and hear from you guys today. Hope you're all doing well. Stay safe and stay healthy. And I look forward to seeing some of you tomorrow and some of you a little bit later. Bye.